Every feature of the human body, from the complex architecture of the heart to this specific pigment of the eyes, relies on biological instructions far too small to see. These tightly wound structures pack the vast majority of our developmental data into a space millions of times smaller than a grain of sand. For decades, we only understood these instructions through mathematics. When Gregor Mendel tracked how traits passed through generations of pea plants, his models relied on invisible factors. He could prove they existed through probability, but he had no way to identify their physical location within a living cell. In the early 1900s, researchers including Theodore Boveri and Walter Sutton began observing cells during division. They noticed that the physical movement of chromosomes perfectly mirrored Mendel's mathematical rules, leading to the chromosomal theory of inheritance. This realization gave abstract genetic traits a concrete physical address. Heredity was no longer just a set of statistical odds. It was a mechanical process carried out by visible cellular structures. But identifying the location of the blueprint was only the first step. The true challenge lay in learning how to navigate its internal map. A chromosome serves as a dense genetic neighborhood. Within a single strand, thousands of distinct genes reside side by side, each governing a different biological function. Because these genes share close physical proximity, they are often inherited together as a single unit. This is the principle of genetic linkage, where the physical position of a gene dictates its likelihood of traveling with its neighbors. Nature, however, shuffles these pairings. During cell division, chromosome strands can overlap and swap segments. This crossing over, or recombination, physically breaks the linkage and creates new combinations of traits in the next generation. By tracking how frequently these genes are shuffled apart, scientists can calculate the physical distance between them. These measurements, recorded in centimorgans, allow us to build a precise linear map of the entire chromosome. Understanding these rules of linkage and shuffling transformed genetics from a series of observations into a precise form of biological cartography. While the nucleus contains the primary map, it doesn't hold a total monopoly on heredity. Some of our traits are governed by systems that exist entirely outside the chromosomal blueprint. Organelles like mitochondria possess their own circular DNA. Because this material is provided almost exclusively by the egg cell during fertilization, these traits follow a strict maternal lineage, bypassing the standard shuffling of the chromosomes. Returning to the nucleus, the chromosomal map handles a different kind of master instruction, the developmental switch that determines an organism's sex. In humans, this is governed by the XXXY system, where a single gene on the Y chromosome triggers male development. In other parts of the animal kingdom, the logic is reversed. Birds utilize a ZW system, where a specific chromosome triggers a female pathway, while bees rely on whether an egg is fertilized at all. Whether governed by maternal mitochondria or the master switch of a sex chromosome, these diverse systems work in tandem to dictate the pathological reality of the organism. Humanity has spent most of its history blindly inheriting its code. Today, we have transitioned into an era where we can actively read and predict it. This mapping has practical, immediate applications. We can now use genetic markers to breed crops that withstand shifting climates and apply precision medicine to identify health risks long before they manifest. Yet this new clarity brings difficult questions. The ability to predict biological outcomes carries the risk of genetic discrimination in our social and professional lives, alongside the complex consequences of selecting for specific traits like sex. Now that the blueprint is exposed, the challenge is determining how to navigate a future where we can see and potentially influence our own destiny. The chromosome is the thread connecting our ancient biological origins to our technological future, a microscopic helix that continues to define the story of humanity.